All right, guys, this is the lecture for part 2 of chapter 13. We're going to talk today about microbial mechanisms for pathogenesis, or how is it that microbes actually cause disease. Now, pathogenesis is the mechanism a microorganism uses to cause disease. Virulence is the relative ability of a pathogen to infect a host and cause disease. It's not related to the severity. So virulence is kind of like how contagious is it? All right, a pathogen's goal is to enter a host, adhere to host cells, gain access to nutrients, evade detection and removal by the immune system in order to colonize, and ultimately leave the host, right? Pathogens have to be able to go from one host to another in order to be effective in their own life cycle. Some factors that affect virulence. How pathogens adhere to host cells is important. There are adhesion factors, so bacteria and viruses have surface proteins that enable them to bind to complementary host receptors. There are adhesions on bacteria, things like pili or physical structures that help them bind and connect to their host cells. There are also attachment proteins on viruses. Some pathogens have a capsule, or they might produce a biofilm. These are all things that helps them stick to the host cells. There are also some host-specific interactions. So adhesins and receptors determine the specificity for pathogens for particular hosts. For example, Neisseria gonorrhoeae. This organism has adhesins on its fimbriae that adhere to cells lining the urethra and vagina of humans. It cannot infect other hosts. They can attach to sperm cells, though, interestingly enough. All right, and then down here you see some examples. These are pili, like kind of linear structures that come out from a pathogen. And here are some host cell proteins in the cell membrane. See the double layer phospholipid membrane down here on the host cell. So these pili are able to make connections with these surface proteins on their host cells, and that's how they make that adhesion or that connection. All right, some traits that influence pathogen virulence. These are like things like how they gain access to nutrients. So they produce extracellular enzymes. They might use these enzymes to dissolve structural chemicals in the body to further invade and gain access to nutrients. They might also produce toxins. We mentioned exotoxins in the part 1 video for chapter 13. These are secreted by a pathogen to destroy host cells. Cytotoxins kill host cells or affect their functions. Neurotoxins interfere with nerve functions. And enterotoxins affect cells lining the GI tract. Each of these three, these are all part of exotoxins. This is a more general term. These three terms here are more specific. Oops. All right, membrane damaging toxins. These can disrupt plasma membranes. Things like hemolysins that lyse blood cells. Another type of toxins are endotoxins, also called lipid A. It's a lipid portion of a gram-negative LPS, and when it's released when G cells die naturally, divide, or are digested by phagocytic cells. It can cause fever, inflammation, diarrhea, and even shock. All right, some other traits that pathogens use to escape detection and removal by immune system are anti-phagocytic factors, things that prevent phagocytosis by white blood cells. So some of the steps might be chemotaxis. So they might move towards certain chemical signals or away from chemical signals. Recognition and attachment, ingestion, or the formation of a phagosome. There's also digestion. So as the fusion of the phagosome with the lysosome And the lysosome, remember, contains chemicals and enzymes that are used to destroy an ingested antigen, forming a phagolysosome. And finally, the last step of phagocytosis is the release of the digested antigen through exocytosis. All right. Some other anti-phagocytic factors are capsules. Capsules are composed of chemicals found naturally in the body, and immune cells don't always recognize them as foreign antigens. Capsules are slippery, which makes it difficult for phagocytes to adhere to them. And sometimes pathogenic cells also use anti-phagocytic chemicals that prevent the fusion of lysosomes with a phagocytic vesicle. A pathogen is able to survive inside the phagocyte if this happens. All right. Other things pathogens might do are avoiding antibodies. Antibodies are Y-shaped proteins produced by activated B cells called plasma cells. They're involved in the adaptive immune response, which means they're specific to their pathogen host or pathogen. And they interact with antigens in a very specific manner, like the lock and key theory, very similar to the way that enzymes and substrates interact. They have an Fc stem and a variable region, and the variable region attaches to the antigen. The Fc stem acts as a flag for phagocytes, allowing for easier recognition and attachment. Secretion of certain proteases break down antibodies. For example, IgA and body secretions bind to bacterial adhesins in pili. Rapid turnover of pili and antigenic variation that can genetically alter the type of pili made, and Fc receptors. Gonorrhea produces chemicals which prevents the fusion of lysosome with phagosome and secretes a protease which breaks down that IgA in mucus. All right, so let's look at a model here. In A, we have a bacterium, or an antigen here, and then this is the antibody here. So an antibody has a specific reaction for an antigen. These two things go together, and that's very specific. The Fab region of the antibody binds to the antigen, so that's right here. And the Fc region of the antibody sticks out like a flag, right? The red part. And this is what phagocytes recognize and bind to this region as the initial step in phagocytosis. So this antigen, this bacterium, has been flagged by antibodies for phagocytosis by the immune system. Let's look over here. B, the Fc receptor on the bacterium binds the Fc region of an antibody. And look, now the flags are all hidden, facing inward, with the Fc or the Fab region pointing out. So this bacterium has outsmarted the antibodies, right? It's evaded flagging for phagocytosis. All right. Now, pathogens also have to leave the current host in order to find a new host, right? It wouldn't be a very effective pathogen if they infected one host and then that was the end. All right, so when pathogens leave the current host, they have to leave through a portal of exit. All right, let's talk now about epidemiology. Epidemiology is a study of the effect of pathogens in a population. So instead of individual medicine, here we're kind of zooming out to like a public health kind of perspective. And epidemiology is the study of where and when diseases occur and how they're transmitted in a population. Why is epidemiology important? Well, the Earth's population is becoming overpopulated. There have never been more humans alive at one time on planet Earth as there are today. And tomorrow, there will be more humans alive on planet Earth than there have ever been at any other time in human history. And that trend isn't slowing, right? So we're going to have more increased public health concerns, and epidemiology is going to become increasingly important as our own population continues to rise. In addition to our own population growth, we as a species are relying more and more on mass food production and distribution. Travel to other countries is readily available. And this leads to a higher incidence or number of new cases of a disease in any population. Prevalence is the total number of cases, new and already existing in a population. So there's some important vocabulary words on this slide. Make sure you understand what these bolded terms are. And if you have questions, make sure you ask them in lab or office hours or with a classmate. All right, occurrence of an infectious disease can be classified in terms of geographic distribution and frequency. So, and we have these listed sort of in order of like smallest epidemiological concern going down with increasing epidemiological concern. They become bigger public health crises the further we go down this list. So, sporadic, endemic, outbreak, epidemic, and pandemic. So let's talk about what each of these mean. Sporadic means a few cases are randomly distributed geographically. For example, plague in the US. And yes, plague like bubonic plague. It has an animal reservoir in prairie dogs, actually. Okay, so these are reported cases of human plagues in the US over a 40-year span. They're randomly distributed in the western US here. So it's not a major public health crisis, but it's definitely something we want to keep track of, right? Okay, outbreak implies a cluster of cases occurring during a brief period of time and attacking a specific population. Often, outbreaks are foodborne. So, for example, E. coli 0157H7 from contaminated food sources would follow this outbreak pattern. Or measles. So in 2019, Michigan had 22 cases of measles originating from a visitor from Israel. All right. And this graph down here is a graph of the distribution of salmonella outbreak. So 77 people became sick and 1 died in a salmonella outbreak caused by contaminated ground turkey in 2011. So you can see it hit hardest in Michigan and Ohio, but there were also cases in other parts of the country. All right, endemic means that it's a native disease that prevails continuously in a geographic region. Endemic diseases can lead to to epidemics. For example, pertussis in the US, or the common cold, or influenza. These are disease that we expect to see in the US, right? Someone with the cold or the flu is not remarkable in the US. These are endemic diseases, and they're prevalent. There is some seasonality to it, although since the COVID pandemic, it seems like the seasonality is not what it used to be. It used to be much more predictable, but still, these are things we expect to find in Michigan, right? They're endemic to our region. All right. Epidemic is a public health concern affecting an unusually large number of individuals within a given region or population. An epidemic may lead to a pandemic. So a good example of this is the H1N1 influenza virus. It was an epidemic in the US in 2009. So look at this graph here is a nice way to compare endemic. The endemic is over time, right? We've got time on the x-axis. The y-axis is number of cases. In endemic disease, we expect to see maybe some fluctuations, but the amount of disease is relatively small compared to an epidemic, right? So even though we get influenza virus every year in Michigan, usually the numbers stay low. But every so often we'll have an epidemic where a certain strain of the flu, like H1N1, shoots up and the number of cases of the disease increases tremendously. All right. Now, I told you an epidemic may lead to a pandemic. A pandemic is when we have an epidemic of the same pathogen on more than one continent. So it's become a global problem. And I remember this because I think of Pangea. Remember when all the mass of land on Earth was in one supercontinent called Pangea? So Pangea is all the land. Pandemic means all the world is having the same major public health epidemic. All right, and a good example of this one is in 2009, that H1N1, or the swine flu, broke out. So it started as an epidemic, but then it broke out and spread to more than 70 countries. So it went from an epidemic to being a pandemic. And if you remember, if it's a swine flu, this means this was an influenza virus that jumped from pigs into humans. See if you can quiz yourself and remember what's the special word we use to describe disease that goes from animals to human. Good, zoonotic. All right, let's talk a little bit more about pandemics. So pandemics is a disease affecting an increased proportion of the population over a wide geographic area, most often worldwide. So because human travel and trade is what it is, usually if something spreads outside of one country, it's not going to stay contained even on one continent, right? At least these things become pandemics, and these are going to be increasingly common as our human population becomes more mobile, right? An example of a pandemic is AIDS. So this figure we have here, it's the estimated percentage of the adult population living with HIV virus infection by country worldwide. So remember, HIV is the virus that causes the disease AIDS. And you can see there are a few countries with very low reported rates of HIV, although I suspect that some of these are probably just not testing for it. But you can see there's blue on every single continent, excluding Antarctica, right? And Antarctica doesn't really count in a discussion about pandemics because there are hardly any resident people there. All right. Hospital epidemiology and nosocomial infections. Especially for those of you going into the healthcare field, this is something you're really going to want to be aware of. Nosocomial infections are hospital-acquired infections, meaning it's a pathogen that a patient picked up while they were in hospital care, or not necessarily in a healthcare setting. You could pick it up from your primary care provider too, or an urgent care. So how do we control nosocomial infections, right? Disinfection, good housekeeping, bathing, sterile procedures, and hand washing, especially hand washing, is the most effective way to reduce nosocomial infections, right? Think of a nurse, on how many patients might a nurse see in a single shift. And if she sees one person with a contagious pathogen, it would be very easy for a nurse to spread that pathogen to all of their patients if they're not being deliberate about doing these practices to control the spread of nosocomial infections. The CDC, the Center for Disease Control, reports that on average, healthcare workers wash their hands before interacting with patients only 40% of the time. That means 60% of the time, healthcare givers do not wash their hands before they interact with the next patient. And that's unacceptable, right? So you will all go out of here and do better. All right. factors that influence nosocomial infections. So there's some things that play a role in what infections are passed around in a healthcare setting. So the presence of microorganisms in the hospital environment, right? The pathogens have to be there to be spread. We have to have transmission of pathogens between staff and patients or among patients. So these microbes are being passed from person to person. And then we also probably have, in a healthcare situation, immunocompromised patients, patients who are already fighting off one infection or some other major health concern. That's why they're in the healthcare setting in the first place, right? So we have a very susceptible population, patient population. And this is when nosocomial infections are most likely to occur. Infection can result from any one of these factors, but usually it's a combination of all three. I think the only one that's necessary is the yellow circle, right? Okay, so we've talked today about how it is that pathogens cause disease and how those diseases are spread. If you have any questions, please make sure that you ask them. You can come see me in office hours, or we can talk about it in lab. But come prepared with questions, because that's an important part, right? All right. Thanks, guys. I'll catch you on the next one.